In this video, we will discuss about this RBPJ act as a negative regulator of FOXP3, and these things they have seen in their previous results also, that we have discussed that RBPJ with the knockdown of RBPJ, it will cause the overexpressions of this FOXP3, and in the literature the effect of this RBPJ has been shown, and it is presented in this panel A, where we can see that when the T cells get activated, this notch signaling pathways will form this NICD, and it will form a co-activator complex with RBPJ, and that will lead to the transcription in a way that will suppress FOXP3 expressions. And when the notch signaling pathway is not activated, then RBPJ form a co-repressor complex, and it will suppress and will act as a repression of the RBPJ, and it will lead to the overexpressions of this FOXP3. And these things they have validated using this volcano plot, where they have analyzed the samples that have RBPJ knocked down and compare it with the control case where RBPJ is not knocked down, or the wild type T cells. And what they are able to see is that in the result we can see the RBPJ has been knocked down compared to the down-regulated compared to the wild type, and the FOXP3 is upregulated. And then thereafter they plotted a heat map where they have seen that some of the genes that have previously been published, that will has an effect with the or that are produced when the FOXP3 is upregulated. That is, the FOXP3 upregulation is on the right hand side of this heat map, and the FOXP3 downregulation is on the left hand side of the heat map. We can see that RBPJ is low in the case of left hand side, and the FOXP3 is high, whereas in the case of right hand side we can see that FOXP3 is low and RBPJ is high. Let me just repeat this thing. So the right hand side of the this heat map shows the condition, the genes that are affected when the RBPJ has been upregulated or it is not been knocked down, and the FOXP3 is downregulated. And the left hand side shows where RBPJ is downregulated or knocked down, and FOXP3 is upregulated. The other genes that are upregulated are shown in the top side, and the genes that are downregulated are shown in the bottom side of the heat map. And they also validate their results using gene set enrichment analysis of the RBPJ knockdown and compare it with the wild type activated T cells. In both cases, the activated T cells are used, and whereas in one case RBPJ is knocked down, in another case RBPJ is present as a wild type. And they are able to see that most of the genes that are enriched is common, or we can see that the enrichment scores are high in that when they compare both these genes in both these conditions. Thereafter, they want to check whether the effect of notch, since in the previous studies results they have not seen the effect of notch, and their hypothesis that it is because of the signaling efficiencies of their method is not good. So when they did the perturbation studies, when the RBPJ is knocked down, they can see the high expression of the FOXP3, but when the other component of the activating notch signals are knocked down, they do not see any significant changes in the FOXP3, indicating that the notch signals do not have a significant role towards FOXP3 expressions. And since their studies involves regularly activating the T cells for the analysis, they wanted to confirm that the results, whatever the results they are seeing, is not the artifact of repeated activations of the T cells. So in that case, in order to take down that possibilities, they use the freshly CD4 plus T cells, and then knock down RBPJ using the oligonucleotide techniques, and they use two different targeting oligonucleotides 1 and oligonucleotide 2, and then activate the T cells and compare it with the non-targeting oligonucleotides. Not able to recall exact ASO full form, but it is some kind of oligonucleotides. And they are able to see that when the RBPJ is knocked down, the FOXP3 expressions goes high in both the cases, in both oligonucleotides compared to the control. And thereafter, there is one more things that happens when the T cells get activated. That is CpG demethylations occurs. That might also have an effect on the FOXP3 translations or FOXP3 protein formations. So they check the CpG demethylations in the case of this control cells activated T cells and the RPJ knockdown activated T cells, and they can see that in the RPJ knockdown activated T cells, the more CpG demethylations occurs, that will gives more stability, and that might have an effect for more FOXP3 formations. So after validating that FOXP3 has a direct role with the RBPJ, they wanted to check with the repeated activations of the T cells, or the activations of the T cells for longer durations has any impact on the FOXP3 expressions or not. So this is how they did the experiment that is mentioned in the panel, left hand side of the panel 7. That is, they activate the T cells and then uh, allow the T cells for the expand, and then again activate the T cells using TNF and then allow it for expand, and then check the FOXP3 signals at day 14 and at day 7. And what they are able to find out in both the donors, they check it for 2 donors, that with increase in time they are able to see the increase in the FOXP3 expressions compared to the day 1, and for both the donors they are able to see the signals like this. And the quantitative analysis is shown on the panel I, where we can see that the comparisons between non-targeting and targeting at day 7, the difference is low, seems to be low, but for the day 14, the non-targeting and targeting difference seems to be high. Although they have not shown the statistical analysis, but from the visual inspection it looks like that. Thereafter, in the panel J, they wanted to check whether the suppression effect of this high activating or high FOXP3 formations on the CD4 plus and CD8 plus T cells, and they can see that when the RBPJ has been knocked down, the suppression effects are high compared to the case where RBPJ is not knocked down for both CD4 plus cells and CD8 plus cells. So this indicates that this FOXP3 not only is negatively regulated by RBPJ, but high expression of FOXP3 also has impact on the suppression of the CD4 plus and CD8 plus T cells. So this is what they have presented in this figure, and they have shown that how this FOXP3 also plays a role towards this immune response. That's all for this video. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.